Bismillahir Rahmanir Rahim. Dear students, Assalamu alaikum and welcome back to Cell Biology, Genetics and Evolution class. In the previous video, we started the discussion of recombination in bacteria, and we saw that there are three basic or simple mechanisms through which recombination takes place in bacteria. And those mechanisms were starting with transformation, conjugation and transduction. So we started the discussion of transformation and the experiments of Frederick Griffith. Now, Frederick Griffith, after performing all these experiments, he concluded, as we saw in the previous video, that the type 2R bacteria, they have been somehow transformed into type 3S. Frederick Griffith suggested that there must be something in type 3S that is responsible for the synthesis of capsule in type 2R, because we know the difference. The major difference between these two strains, for example, these are the Diplococcus bacteria. So this is the difference, the capsule in type 3S and no capsule in type 2R. So these are R type and these are S type. So obviously he knew that these R type bacteria somehow developed the capsule. And what he said, that there must be some substance, a chemical or something in dead type 3S cells which caused the development of capsule in type 2R and which transformed them into type 3S. And he named that substance or chemical or whatever it might have been transforming principle. So these were the conclusions of Frederick Griffith. So he said that there must be something, a substance, a chemical, a compound, the transforming principle in dead type 3S cells, and this transforming principle transformed or converted R type into S type. Now, after the discovery of Frederick Griffith, some other scientists also performed these experiments. In fact, they confirmed the discovery of Frederick Griffith. So the first person was Henry Dawson. Henry Dawson of Rockefeller Institute. Now he performed experiments in 1931 and he confirmed the discovery of Frederick Griffith. Now Henry Dawson and his co-workers not only confirmed the discovery of Frederick Griffith, they in fact extended it to one more step, and that is they showed that the transformation of type 2R into type 3S, it can be carried out in vitro, that is in test tube. So it can be carried out in vitro as well, in test tube. That is, if we mix the two cells, type 2R and type 3S, in a test tube, the type 2R cells ultimately get transformed into type 3S. And they showed that all that was needed was incubation of these two cells, type 2R and type 3S. So incubation of these two cells together, incubation of type 3S, and obviously they are dead or heat killed, plus living type 2R. Type 2R, living type 2R. When these two cells are incubated together, after a certain length of time or incubation, live or living type 3S type cells can be recovered. So these were the experiments of Henry Dawson and his co-workers who confirmed the discovery of Frederick Griffith. Another scientist, Lionel Alloway, he further refined these experiments, and instead of using cells heat killed type 3S, he used crude extract. So instead of using cells, he took crude extract of heat killed type 3S. So these are heat killed type 3S, and a crude extract was taken, and this crude extract was mixed with living type 2R, and he obtained the same results, that is transformation. These type 2R, they were transformed into type 3S. So the experiments of Lionel Alloway confirm that there must be, and these experiments were performed in 1933, about 5 years later after the discovery of Frederick Griffith. Now Lionel Alloway proved that there must be some chemical because he used extract, so extract only contains chemicals, so there must be some chemical compound in that crude extract taken from dead type 3S cells. So this chemical was responsible for the transformation. However, there was no satisfactory answer as to what is causing the transformation. After all these experiments, first Frederick Griffith, then Henry Dawson, and then finally Lionel Alloway, demonstrated that there must be some chemical responsible for this transformation. So the question was, what is transforming principle? What is the chemical or that chemical which is causing this transformation? So this was proved by, or the answer was finally found by, a team of workers led by Mr. Avery, McLeod and McCarty. They performed a set of ingenious experiments, fascinating, superb experiments, in which they finally found out the transforming principle. In fact, they exclusively confirmed it, that what is the transforming principle. So we are going to take a look at a figure, a diagram showing their experiment. We are not going to go into details of their experiments, and then we are going to describe that how transformation takes place once it was found that what is the transforming principle. So let's see in a diagram or figure the experiment of the team of workers Avery, McLeod and McCarty. So this is a diagram showing their experiment. So as you can see, what they do, that they took type 3S cells in a culture medium, centrifuged it, and then heat killed it. And then what was done? Homogenization of the filtrate. Please don't worry about all these steps or how it is done. What they were trying to do was to collect the extract. So they collected the extract. Now this is the crude extract which we have just mentioned. If you see here, Lionel Alloway, he obtained the crude extract, and he could duplicate the results of Frederick Griffith with the extract. So this is what these scientists were doing, that they took the extract. So hopefully you are following me. Next step was to find out which compound in this extract is responsible for the transformation. That is, what is transforming principle. Now, as you know, when you take extract from a cell, so it contains all these compounds which you have already studied before the mid-semester examination, the biomolecules, that is proteins, carbohydrates, lipids, and of course nucleic acids. So the extract contains all the compounds. So their job was to find the transforming principle. Obviously, one of them, these carbohydrate, proteins, lipids, and nucleic acids, one of these compounds was responsible for the transformation, or one of these was the transforming principle. So this is the extract. What they did next was to treat this extract with various chemicals to find out the transforming principle. So they took the extract, mixed it with the type 2R, and what happened? Transformation occurred. So this is their control experiment. I repeat again that they took the extract, filtrate, and mixed it with type 2R, and transformation occurred. Simple. Now, this row tells their treatments with these extracts, what they did. So what they did, they treated the extract with protease. Now what protease does, that it removes all the proteins. So they treated this extract with protease. Now it does not contain any proteins. Then it mixed this with the type 2R, but the transformation occurred. It means the proteins are not responsible for transformation, because the protease test or the protease treatment removed the proteins, but transformation occurred. As you can see, transformation was found to occur. Next, they treated this particular, which there were no proteins, treated them with ribonuclease, removing the RNA. So after removing the RNA, they mixed them with type 2R cells, but transformation occurred. That means that RNA was not responsible for the transformation. Next, they treated the extract with deoxyribonuclease, the enzyme that destroys DNA, and they found the answer. When they treated the extract with deoxyribonuclease and mixed it with type 2R cells, no transformation occurred. Only type 2R cells were found in the filtrate. Now this was proved conclusively by this team that the transforming principle was DNA, and it was neither carbohydrates nor proteins nor any other compounds, lipids or RNA. So they obviously it was a huge experiment, and it took several years to prove. In 1944 they published their results, almost 10 years, after which they found out that the transforming principle was DNA. So it was finally proved by this team of workers, Avery, McLeod and McCarty. It is their experiment, that the transforming principle is DNA. So after having found out the transforming principle, in the next video we are going to take a look at how transformation takes place, how a type 2R cell is converted, transformed into a type 3S. So that is our topic of the next video. Thanks for watching, and see you in the next video.